Okay. Hello, everyone. I am going to go ahead and do this review for you all. Um, let's just get started, just jump right into it. Okay, so let's start off with what we know, the cell cycle. Of course, cell cycle has a few different parts in it. We could divide it up in a few different parts, right? We start in G1, we have an S phase, and we have a G2 phase, and then we have an M phase, and within M phase we have a few different other phases, right? So G1, within G1 we have, you can consider G to be a couple different things. You could consider it to be growth, or you can consider it to be a gap, right? So it stands for gap, right? But primarily it is during this time where a cell just performs normal cellular activity. It is a gap between the previous mitotic phase and the following synthesis phase. So gap just meaning like, the space, that space, right? And within that space, that cell does normal functions like growing and like, I don't know, just doing normal metabolic functions. That's what G1 is, right? Moving on into S phase, that's when the DNA within the cell actually goes ahead and duplicates, right, or replicates. You could use those terms interchangeably. It doubles, is essentially. So G1 into S. During S, we have minimal cell function, but there still is some cell function. However, it is during S phase that all of the DNA replication stuff actually takes place. It's S for synthesis, not S for super, it's S for synthesis. Right? Synthesis into G2. Now, G2, we have our chromosomes that have been duplicated, right? And G2 is essentially just the cell doing normal functions, but also getting ready for M phase, which is, of course is the mitotic phase, in which we have a few different phases like prophase, prometaphase, anaphase, telophase, and then we ended off with cytokinesis. So the mitotic phase is essentially just when the cell finally decides, okay, I'm going to make another one of myself now. So you take one cell and you turn it into two identical cells. Right, so that's the basic cell cycle. Let's go into a little bit of S phase. So, with S phase in mind, we wanted to take a closer look at the DNA. Right, so this is your DNA. It's of course, the double helix. Kind of looks like rungs on a ladder, right? So that's our DNA. And one thing that's really important to note before we even begin to jump into this, right, is the base pairing rules, right? So of course we have A, T, C, and G that we all worry about. These are our four nucleotide bases, right? So adenine, thymine, cytosine, and guanine, right? A, T, C, and G. And what essentially base pairing rules are that we pair A with T and C with G. So always these letters are going to pair with each other. And that's the case for DNA, right? So DNA, of course, has two strands, right? So this is the case for DNA. The case for RNA, C remains with G, but A now binds with U. And the way they do it is through hydrogen bonding. Not through covalent bonding, but through hydrogen bonding, right? So, with that in mind, we can now go ahead and continue on with our discussion of DNA replication, right? So, if we think about DNA, right, you can kind of think of it like a zipper. So, if you think of a zipper, you could unzip it and you could zip it back up again, right? It's a reversible process, but if you want to open something, you open up the zipper, right? You want to get to what's inside, and then you just close it back up again, right? In the same way, DNA can be unzipped and rezipped. So let's go ahead and draw our DNA that's been unzipped. So this is our unzipped DNA. Now we can see that the rungs of the ladder, so to speak, have been undone, right? Now, this doesn't just happen by itself. There has to be the aid of an enzyme, and I like to draw that enzyme as a little triangle that's going down the middle here, right? So this middle portion, this spot right here, is known as the replication fork. So that's important to note, right? The replication fork. That's kind of where all the magic happens, right? As the helicase, oh well, there's your answer, right? As the helicase is going down the replication fork, it is unzipping this DNA, and it is allowing for a space for DNA polymerase, which is going to add more nucleotides. It is going to create a space for those to go ahead and do their thing, right? So, with that in mind, we now have an unzipped DNA. So what do we do with this, right? So by the way, right, just as a second note, this enzyme, so an enzyme is a biological it is a biological protein that helps perform a function. In this case, helicase is a protein that is helping unzip DNA, and that is a very basic and not entirely accurate definition of an enzyme, but for now it'll have to do. So this is a helicase, right? So helicase goes ahead and unzips the DNA. So now we have two exposed strands of DNA. And of course, if you remember correctly, you'll remember that DNA has two ends to it, right? So DNA has this base sugar, and then it kind of, and then it expands off into the, base, right? Into the nitrogenous base. Let's put an N there. So that could be, that's what makes it different between A, T, and C and G. That's the base, right? Base can differ between these different nucleotides. And then we have the phosphate end, and we have the 3' hydroxyl. So with all of that in mind, right? With all of that in mind, we have our basic nucleotide. So with the nucleotide, we could actually count the number of carbons: 1', 2', 3', 4', 5'. And the reason why they're primes and not just the numbers is because of the carbons over here in the nitrogenous base. Those are the ones that are without the prime, but the one in the center are the ones with the primes. So with that in mind, we care about, oh, I did a mistake. That's not where the 5' is. It's up here, actually. Because normally, this is actually an O. This is actually an oxygen that helps bridge together the the sugar. So the 5' end is up here, and the 3' end is down here. Basically, that's, if you ever hear 5' or 3', that's what's meant here, right? So this 3' hydroxyl group is really, it loves to react with things. So basically, this 5' group does not like to react with things. So with that in mind, when we're adding nucleotides, we're always going to add to the 3' end. So we're always going from 5' to 3'. 5' to 3'. That's always the case. Whether it's for RNA or it's for DNA, it's always from 5' to 3'. So that's just how it looks in general. I don't think you really have to know what the actual molecule looks like, but it is important, in my opinion, for that distinction. Right, so let's move on. So that's our DNA molecule, and what that DNA molecule looks like. So let's consider this end to be our 5' end, because remember, DNA runs anti-parallel. So we have one strand running this way, and another strand running this way. So they're parallel, meaning that they're never going to intersect, and they're anti-parallel, meaning that they're going to not only not intersect, but also be running the opposite direction. So on this end, we have the 3' end, and on this end, we have the 5' end. So with that in mind, we now have the basis for how DNA is going to be added, right? So, DNA by itself cannot just be added with it can't just be added. We have to have a special something, a special like a start to it, right? If you think of like a primer, right? If I think of primer, I think of like a gun, right, or like a bullet, right? The bullets just don't go off by themselves. You have to pull the trigger first, but for one, you have to have a primer, and the primer sets off a little explosion that sets off the gunpowder that makes the bullet go flying, right? In the same way, we have to have a primer on the DNA to facilitate, or not only to facilitate, but to begin replication, to kind of kick it off, right? So this primer, let's make it orange, is added, right? And this, let's actually not orange, let's do blue. So this primer is added by an enzyme known as a primase. So primer, primase, primase adds a primer, right? So these primers are going to be made of RNA, right? So later they're going to have to be removed, right? So with that in mind, we put down a primer, and now that we've put down the primer is actually going to face the opposite direction from the DNA strand it's originating on. So on this end of the primer, it's going to be 5 prime, and on this end, it's going to be 3 prime, right? So the direction, so now that we have the primer, the direction of the DNA synthesis is going to be within that direction. So now that we have the primer acts as a signal for a certain other enzyme known as DNA polymerase. Let's make that red. It creates space for a DNA polymerase. It doesn't create space, it just creates a sort of signal for DNA polymerase. So now that we have DNA polymerase is just going to go ahead and it's just going to go ahead and go, right? That's it. That's an example. That's an artist's rendition of how that's going to work. DNA polymerase is just going to go as the helicase unzips. It's just going to go, right? So with everything that we've said in mind, we can now proceed with how this is going to occur, right? So DNA polymerase creates DNA nucleotides in the 5' to 3' direction. So if you remember, down here we have the 5' end, right? So that's the direction the DNA's that's the direction of the DNA, right? So it's going to be the opposite direction up here. So it's 5'. So that means that we're going to make the DNA in this direction, right? So DNA polymerase is making 5' to 3', and there's no problem. There's no problem. It just goes, right? 5' to 3', just making that DNA, and there's like I said, there's no problem. However, there is a problem when we get to the other strand of DNA, right? So we still are running from 5' to 3'. So now 5' is on this end, and 3' is on this end of the primer, right? So now we're going to go ahead and take the primase again, and we're going to move it. It's going to move, right? So now it's going to move this direction. And it's going to make a primer here. However, notice how the replication fork is unzipping. The helicase is unzipping more DNA at the replication fork. So how do we account for this? Well, we just add another primer. And when we add another primer, we just add more DNA. Add another primer, add more DNA. It's really that simple. However, right now we just have a bunch of primers that need to be removed. They need to be excised, right? So, with that in mind, we take a exonuclease. Let's make that pink. Exonuclease. And this thing comes in, and it kind of removes the primers, right? It removes them. So we make them from blue into white. Sorry if I just blinded you. I did it again. However, there is a bit of a problem, as you may see. There are little tiny gaps that are created here. And those gaps need to be filled in. And by the way, there's something that I need to mention, right? These so-called fragments are known as Okazaki fragments. Okazaki fragments, right? Some Japanese biologist named Okazaki, discovered these fragmented DNA pieces, and during the S phase of DNA replication, during the S phase of the cell cycle, and discovered that, well, there is that discrepancy between the two strands. With that being said, also the strand that's going within, the strand that's going into the replication fork, that is the leading strand. So remember that. Into replication fork is going to be the leading strand. Now the opposite is true. If we're going away from the replication fork, that is the lagging strand. So into the replication fork, leading. Away from, lagging. Right? And when we have the lagging strand, we have these Okazaki fragments. So like I said, these Okazaki fragments do need to be sealed back up so we have a uniform strand of DNA. Right? So once we have the enzyme, once we have these little gaps, we have an enzyme, and that enzyme is known as ligase. So ligase makes a ligand, meaning it connects two pieces. The ligase comes in, and it makes this uniform strand of DNA, just like that, right? Just like that. So now we have two identical strands of DNA from one original strand, right? So we have one original strand, we split it in two, and from those two bits, we make more DNA. This process is known as semi-conservative. So semi-conservative means that we have the original bit, and we keep a little bit, we keep like half of that original bit, and we build off of it. So semi-conservative, right? So that's the basics of DNA replication. Now just imagine this entire process, right? Just imagine this entire process. Let's see if I could do this. That didn't work as well as I thought. Okay, wait, hold on. Ah. I really believe in this working, by the way. You can't tell. Okay, well this isn't working. Okay, well we're just gonna ignore that for now. Okay, just imagine this happening on the other side, right? The other side of the quote unquote replicate the other side of the DNA, right? So I'm just gonna pull up a picture of it because that's easier. I'm sorry this picture is so bad, but so basically we're just looking at the left-hand side of this image, right? So this is the replication bubble, right? And we were doing what we just described on two sides of the same strand of DNA, right? So here we have a helicase that's unzipping. over here we have a helix that's unzipping, and etc., etc., right? So, with that in mind, on this, so we have the leading strand that's going in here, but then on the lagging strand, it's cut up in little bits, right? But they're both going the same direction. So notice how that kind of changes, because over here we're going into the replication fork, and over here we're going away from. So now this one's the leading strand, this one's the lagging strand, but they're both on the same strand, right? So that it can change, right? So same thing down here, where we have these little fragments, and then it goes continuous, kind of like that, right? Okay, so imagine that. Imagine everything we just learned, and that happens during the S phase of the cell cycle. So remember that, the S phase of the cell cycle, that's when all of this stuff happens. So, for example, the enzyme, so as a quick review, right, the enzyme that unwinds and separates the DNA double helix is, of course, going to be the helicase, right? Remember that one. The leading strand is going to be going into the replication fork. Lagging strand is going to be going away from, right? But remember, they're both being made from 5' to 3'. I think I didn't. So DNA polymerase is going to do all the magic. It's going to add more nucleotides. But remember, it's always going from 5' to 3'. And also remember, DNA polymerase cannot just do this on its own. It needs a primer. It needs a primer. So we need the primase in order to actually start this process. Primase, primer, and then finally we could put in the DNA polymerase to actually do it, right? So consider the leading strand and the lagging strand. Leading strand is going to be continuous. Lagging strand is going to be discontinuous. Makes enough sense, right? And all of this all of this action happens during the S phase. So DNA polymerase does this work, and yeah. So what does this all mean anyway? That's not supposed to be a existential question. I mean, literally, what does it all mean? What is the purpose of this? Why are we talking about this? Well, when we make a cell, we want the two cells that arrive from one cell to be identical to each other. So, in order to do this, we have to have double the amount of information we started with, right? So now we're going to shift discussion into the talk about chromosomes, right? So chromosome is basically just a little packet of information, a packet of genetic information that's neatly packed together, neatly coiled, has a certain number of genes, and can be transferred from generation to generation, right? So, for example, humans have 46 chromosomes, right? And humans get 23 from their mother and 23 from their father, right? We'll go into why that is later, but just for now, know that 23 from mom, 23 from dad, right? And we put them together, you get 46, right? So how did this happen, right? Why do you have 46 chromosomes? Why is that important, right? Well, your chromosomes contain all of the genetic information. It contains all of the information, all of the directions, all of the blueprints to make another one of itself, right? And to specialize, not only. I mean, think about your body, right? Think about your brain cell, and think about your pancreas cell. Two very different cells. Or think about the cells in your eyes, right? And think about the cells in your stomach. One makes acid, and one makes you see, right? However, all of these cell types have the exact same set of genes. And that could be attributed to the fact that the chromosomes are all the same and all kind of performing different functions, right? So chromosomes, we have a couple of different ways of keeping track of chromosomes in the human body, right? And that's with this letter, N, right? So N tells us the number. N is the set, or the number of sets of chromosomes. Sorry, my cat is really making this hard. So N represents the number of sets of chromosomes, right? So with that in mind, right, Humans, like I said, have 46 chromosomes, but because you have two sets, one from dad, one from mom, we now say that 2n is equal to 46, right? Or maybe not number of sets of chromosomes. Maybe that's kind of inaccurate. That's kind of misleading. Well, this number right here, 2, actually represents the number of sets. N just kind of represents the number of chromosomes, right? So not number of sets of chromosomes, just number of chromosomes, we could say. And this 2 can represent the number of sets, right? So 2n equals 46. I mean, obviously, if you know basic algebra, you know that we can simplify this to say n equals 23, right? So n equals 23 is what a sex cell would contain, for example, right? And we have a couple of different words for these. 2n is a diploid cell, and n is a haploid cell. So your diploid cell is going to be every single cell in your body that has a nucleus that is not your sex chromosomes. That is not your sex cells. Haploid is going to be exclusively your sex cells. So, like in the case of you being male-bodied, that will be sperm, and in the case of you being female-bodied, that will be an egg cell, right? So we have diploid, and we have haploid. We take two haploids together, and we make a diploid, right? So n represents the number of chromosomes, right? So you may have seen a chromosome, let me look up a karyotype for you. So this is a karyotype, and I'm sorry if it's a really, really just terrible image. That's on me, and let me see if this is any better. Okay, see, this is better, right? So this is a karyotype, right? A karyotype shows you the number of chromosomes and what's going on maybe, and what kind of genes you have a little bit, right? You just slice open a cell and you just pick out the chromosomes that you want to examine, right? And as you may notice here, we have 23 pairs, right? So 23 pairs, that gives us 46 chromosomes. And as you may notice, we have this pair, which is similar to each other, and we have this pair, which is similar to each other, and vice versa, right? All until we hit the X chromosome, right, which are the sex chromosomes. So if we look within this one pair, right, this pair is very similar to each other, right? We have this little pink regions and this little green region in the middle, and these little lime green regions on the ends, right? We take these patterns, does 3 have those? No, not really. How about 10? No. 16? Not even close, right? So these pairs we consider to be homologous chromosomes. So these pairs are homologous chromosomes, meaning that they contain similar content, about the same length, the same amount of base pairs, but they are ultimately a little bit different because you get one set of your homologous chromosomes from dad and one from mom. So we can consider this one, for example, this one on the left, to be from mom, and this one on the right to be from dad, right? Just like that. And the more genetic stuff about it we'll go into later, just basically basic inheritance, right? If your dad has blue eyes and your mom has brown eyes, chances are you're probably going to have brown eyes when you're born, because blue eyes are considered to be a recessive or not as frequent trait as brown eyes are. But in any case, that color for eye, the gene for eye color is located on the same spot of the chromosome in the homologous pair. So we have the gene for eye color here and here, for example, but not here and here, because that will exist here on the number 1 pair of homologous chromosomes, right? So with that in mind, we have the homologous chromosomes, and we have, yeah, that's it. We have the homologous chromosomes. But when we get a little bit deeper into it, we'll know that's not necessarily always the case. Well, it is the, oh my gosh, see, it's kind of early in the morning or late at night, depending on how you want to look at it. I'm sorry if I am rambling or seem a little insane. Yeah, I hope you will get something out of this video. In any case, we are going to continue discussion about chromosomes. So, we have these 2 pairs, right? So we have these chromosomes that kind of have the same length. This is our cell, or this is our nucleus, and this is our cell, right? So within our nucleus, we have this amount of chromosomes. If we count them, we have 1, 2, 3, 4, 5, 6. So if you're keeping track at home, you will know that the cell is 2n, right? Because it's a diploid cell, because we have homologous chromosomes. 2n equals 6, right? So you have 3 pairs, all containing 2 chromosomes, right? So this is a pair, this is a pair, and this is a pair, because they all have the, like I said, the same amount of information, similar content, but ultimately they are different, right? So they're a pair, but they're not the same. So if you think about like pair of twins, for example, like a pair of identical twins, I mean, ultimately they're gonna have certain differences, but they're almost the same, right? So this is what a cell looks like in, for example, G1 phase. So if you remember earlier, G1 phase is our gap phase, right? Our first gap phase. And then we go through S phase. So during S phase, we duplicate all of our chromosomes. So now our cell is going to look a little bit different. The nucleus inside of our cell is going to be a little different because we have now doubled the amount of DNA, right? So what does that look like? So now instead of these straight lines, we have these sort of X's, right? So let's take a closer look at these X's in particular, right? So this pair of X's is going to be a pair. Of course, this is going to be a pair. This is going to be a pair. This looks kind of familiar, doesn't it? That's right, because we still have homologous chromosomes. We still have the homologous pairs after S phase has occurred, right? But now, if we zoom in a little further, we will see that instead of just 1 chromosome, we now have 2 chromosomes. Except that that's not really the case. There are not 2 chromosomes. This is still considered to be just 1 chromosome, right? But if it's 1 chromosome, then why are there 2 of them? Well, that's because these little constituent parts are known as chromatids. Specifically, they are sister chromatids. Sister chromatids. So in this case, sister chromatids means that these 2 chromatids are exactly the same. So if you remember, homologous chromosomes are not exactly the same. They are not identical to each other. Sister chromatids are identical to each other, right? Very important distinction, right? So this one right here and this one right here going to be 1-to-1, exactly the same, right? However, they still reside within 1 chromosome. So if we take the other chromosome, now how many chromosomes do we have? Do we have 4? No, we have 2. But we have 4 chromatids. So we mark it down. 1, 2, 3, 4. 4 chromatids. And within this, we have a pair of homologous chromosomes. So this is 1 chromosome. This is 1 chromosome. Within each chromosome, we have 2 chromatids because they are identical to each other. And between all of this, we have 1 pair of homologous chromosomes. So that's a lot of vocabulary, right? Just remember, just remember, when you see this little X, that is not two chromosomes. It is two chromatids, but it's just one chromosome. Two chromatids, one chromosome. So our homologous chromosomes. So I have a little question for you to see if you were hopefully paying attention. Are homologous chromosomes identical? No, they're not. Are sister chromatids identical? Yes, they are. How many chromatids are in a pair of homologous chromosomes? 4. How many chromosomes are in a pair of homologous chromosomes? 2. Right? So this cell doesn't change, right? This cell is still 2n equals 6. Right? But we're going to see later why this distinction between this like sort of X conformation of a DNA of a chromosome, this little X conformation of a chromosome is different from this sort of like line conformation. Right? So the DNA has been duplicated, right? The DNA has been doubled. We know that. But what does this mean? Why does it matter? Right? Why does it matter we have X's, right? Instead of just lines. So with that in mind, we are going to move into cell division. Right? So cell division has a few different steps. We have prophase, prometaphase, metaphase, anaphase, telophase, and cytokinesis. So let's start before prophase. Let's start with a cell during G2 phase. Right? So let's start with a cell that's, for example, 2n equals 4. So this cell is going to have its DNA molecules, but it's going to be all kind of like spaghettified like this, right? Just imagine all the DNA kind of like in a loose and not organized fashion. So this is during G2. So we don't really care about G2. It's just an example, right? Or ignore that. We do care about G2. So we have this cell before division, and now when we enter division, we enter into prophase. Let's call it P. Well, now we enter into prophase. Now a couple of things happen, right? We still have this sort of central. We still have the nucleus, but now instead of being like a spaghettified like this, now we have our distinct chromosomes. And these are considered to be condensed, right? So during prophase, we are condensing the chromosomes. So not only that, but we're starting to see the formation of these organelles on the poles of the cell, known as centrioles, known as centrosomes. Right? So these centrosomes are going to do some magic later about binding some spindles to the chromosomes and everything, right? So this is going to be prophase. So now we're going to move into prometaphase. So now during prometaphase, we have the nuclear envelope. So this is the nucleus that's being dissolved. We have the nuclear envelope being dissolved, and we have our chromosomes that are condensed. So that's from the previous step. But now we have our centrosomes, and our centrosomes are beginning to attach these spindles to each of the chromosomes. So these spindles are going to go ahead and facilitate all of the movement of the chromosomes, right? We want to make sure these chromosomes are all organized and all accounted for, right? So basically, one quick little note, this is important, please do remember this. We have a chromosome that's been duplicated. Within this chromosome, we have two sister chromatids. Now, how are these sister chromatids attached to each other? In the middle, we have what's known as a centromere. Centromere, right? And these spindles know, so at the centromere, these spindles know where to attach, right? And at the end of these spindles, we have kinetochores. Now, that's not something I'm 100% on. That's not something that I'm super confident on. But it is still important to know what a kinetochore is. So a kinetochore is going to kind of take its little arm and attach to the centromere, and it's going to facilitate movement of the chromosome, just like that, right? So this is prometaphase. So we have prophase, and then we go into prometaphase. So now it gets a lot easier, right? So after prometaphase, we have, of course, metaphase. So during metaphase, we have the envelope that's fully dissolved, and now we actually have our chromosomes that are lined up perfectly along the center. And this is called like the metaphase plate or something. Not super important. So we have these centrosomes that have gone ahead and bound the spindles to the center, to the centromere, and this is going to be metaphase. Right, so at metaphase, that's when the chromosomes are aligned at the center, primed and ready for splitting, right? So now we're going to move on to anaphase. So for anaphase, now we have the DNA. We have all of the DNA that's migrating to each pole. Now we're migrating towards each side of the cell, right? And at this point, and I know this is going to be very confusing, so bear with me. At this point, these chromatids are now considered to be their own chromosomes, right? So after these chromatids have been split apart from their they are now considered their own chromosomes. It's weird, I know, but something just we have to deal with. So anaphase, this is anaphase. Now we're splitting apart the chromosomes. We're splitting up the sister chromatids. They're now going into their own cells. They're saying bye-bye to each other, going to their own cells, and we're going to do that with telophase. So with telophase, we form what's known as a cleavage furrow. This is a cleavage furrow, kind of looks like a butt, right? And we're going to undo everything we did in prophase and prometaphase, right? We're going to undo everything. So now we're going to decondense the chromosomes. Notice how I'm drawing 4 chromosomes still. We're going to decondense them, and we're going to kind of pinch off using well, I don't quite remember, but we're going to pinch off the cell membrane here at the center. We're going to pinch it off, and we're thus going to create 2 new cells. So this is telophase, and then we move on to cytokinesis. We now make two identical cells. So notice how these ones down here are pretty much exactly the same as these ones up here, right? This would actually be a little misleading. Let me just draw it for you. Okay. So now, after telophase, we have the process of cytokinesis. Cyto meaning cell, kinesis meaning movement of. So cytokinesis, movement of cells. We're splitting apart this cleavage furrow into 2 different cells. Both are identical, and both are identical to no, okay, not only are they identical to each other, but they're identical to the parent cell, which is this one right here, right? So that was cell division. We start at G2 phase, and then we go into prophase, where we condense the chromosomes. We have these little centrosomes as well, and now we're starting to in prometaphase we're starting to form the spindles. We dissolve this we dissolve this nuclear membrane, and then we move these chromosomes to the middle for metaphase, and then during anaphase the chromatids are split apart, and then telophase we undo everything we just did. So we're going to recondense the we're going to decondense the chromosomes, and we're going to reform the nuclear membrane, in which after the cleavage furrow is split during cytokinesis, we are going to have two identical cells, not only to each other but also to their parent. Any questions? Obviously, no, because you can't talk to me through the screen. But in any case, these are the steps of mitosis, right? So if you remember earlier of why I was making the distinction between this type of chromosome and this type of chromosome, is because this type of chromosome cannot be split apart, right? It cannot be split apart, right? So if we didn't go through cell division, then these last cells would have half the amount of DNA as the parent, right? That's very important to note, right? And if you look at these ones, so now that we have two identical copies, we can now put one copy into one cell and the other copy in the other cell, therefore creating two identical copies. Do you see why that's important now? I hope you do. In any case, we are going to now move on from the discussion of mitosis into the discussion of meiosis. Whoops. My cat's making this a little bit difficult for me. Once again. Um, yeah. So just as a quick review, right? When we divide the cytoplasm, when we divide the cytoplasm, that is cytokinesis. That is the definition of it, the division of the cytoplasm, right? And the movement of cells. So also, when we have condensed chromosomes and the nuclear envelope is starting to disappear, that's how we know when we're in prophase, right? So, and also, cells undergo DNA replication because they want to double the amount of chromosomes that they have. They want to double the amount of chromosomes that they currently have. They want to double it so they could make an identical copy later, right? And as for why cells undergo mitosis, well, it's for growth of an organism, right? It's for growth of an organism. If you're a bacteria cell, it's for asexual reproduction, and if it's you, that's the replacement of rapidly dividing cells and rapidly dying cells like skin cells or like your stomach cells, I think. Cells that are rapidly being shuffled in and out, right? So regarding sessile chromatids as well, they have identical DNA sequences. We know that. On the chromosome, we have on the double chromosome we have like the little centromere, and the centromere attaches the kinetochore. Right, sister chromatids is separated at anaphase. Um, yeah, yeah. So it's a yeah. Sorry, I'm starting to feel a little fatigued from all this. Um, cool, cool. Let's go over meiosis real quick, and Let's talk about energy, and then we'll be on our merry way. Okay, so let's go ahead and just start it up. Whoops. Okay, so so now we're going to talk about the production of sex cells. So sex cells, gametes, sperm and egg, however you want to call it. Sex cells are going to have half the amount of information that a normal body cell is going to have. And the reason for that is that because, like I said earlier, when you combine two gametes, then you get a fully fledged organism, like you. But in the meantime, how do we make these sex cells, right? So we start a cell in G2, just like how we do in mitosis. Now we're going to a process called meiosis. So we have mitosis, like mighty, like making a new organism. No, like making a big organism bigger. And then we have meiosis, which is like meiosis. Like, how do I make me? How was me made? So meiosis, right? So we're in G2, right? That's pretty standard. And now in G2, so now after G2, we're going to, instead of going to prophase, we're now going to go into prophase 1. So that's already a major difference, right? Prophase 1, what does that mean? Well, it means that we're going to go through meiosis twice. There are going to be 2 cycles of cell division. Right, so prophase 1, right? So during prophase 1, we're just going to do, we're going to do all the stuff. We're going to do all the big hits, right? We're going to condense the chromosomes, and just like earlier, let's say 2n equals 4, right? We're going to condense the chromosomes, except now it's going to be a little bit different. Right? It's going to be a little bit different because now we have to make the distinction between chromosomes you got from dad and chromosomes you got from mom. Right? So let's make mom red and let's make dad blue. Now, the chromosomes are actually going to interlock with each other. And this is really important for a reason I'm going to go in a second. They're going to interlock with each other. Right? And not only that, but we're going to have the centrosomes that are going to attach, right? Going to attach and everything, yeah. So, basically, what's happening right here? What's happening at this sort of intersection point? Well, let's look closer, right? At these intersection points, this is known as crossing over. So now crossing over is when 2 chromosomes interchange genetic information, right? So if you take genetics, you'll know this is like recombinant gene therapies or whatever. But for now, let's just discuss crossing over. So crossing over is like when you have 1 chromosome and you have another chromosome of the same pair. So if we have a homologous pair of chromosomes, they interlap with each other and they swap information. So this process creates genetic diversity because you're never going to, or you're almost never going to have 2 chromosomes as a result of this that are going to be identical with each other. So basically the end result of this is that we have 1 chromosome and then we have another chromosome. This red one, the one from mom, has certain bits of blue in it, right? Now they have certain bits of blue in it. And the one from dad has certain bits of red in it. Right? So notice how this is different from the original product. This is different from the original chromosome, right? Because this is due to a process called crossing over. This only happens during this phase, during prophase 1. Only, and that's important to note. So, with that in mind, now what do we do with all this information, right? With that in mind, this just creates genetic diversity. That's the end goal. Create genetic diversity. I mean, have you ever wondered why you look so different from your parents? You ever wonder why that's important? You ever thought if you're, for example, if you were, if your grandfather had a genetic disease and your paternal grandfather did not have a genetic disease, but your maternal grandfather does. Well, wouldn't it be better if, I don't know, you had your mom and your dad, and you would have a much lower risk because your mom does not have that disease or is not a carrier of, but your dad is. It would be a lot better if you weren't, right? So if your mom didn't have that gene, but your dad did, right? You as an individual would have a much higher chance of that gene kind of not being expressed, right? So genetic diversity is like the seed of Darwinism. It's the seed of evolution, right? This change, this cause for change, this need for change helps us thrive, and it helps us not be just eliminated by some super deadly virus, right? So yeah, so yeah, so this is all happening during prophase one, right? So prophase one, just imagine prophase and prometaphase all condense into one step. So now we're gonna go ahead and move into metaphase one. So during metaphase one, now we're going to, and I know I kind of drew it up here like that, but I didn't really mean for that. Now during metaphase one, we're going to align, and this is another big difference, right? We're going to align the homologous pairs. We are not going to align them one by one by one by one. We're going to align them by pairs, right? So now we have our chromosomes kind of aligned like this. Right? Now, there's another little bit of genetic diversity that could occur here, right? So once these split off, we then have one cell with all mom's genes and one cell with all dad's genes. But look at this, right? We have another little tidbit, another little thing of genetic diversity to be had here. So what if instead what if now we had dad's genes here and here and mom genes right here, right? So when we split apart, now we have a cell with mom and dad and a cell with mom and dad and mom, a cell with dad and mom, right? Do you see that distinction? Right? Like this process will therefore create different, therefore contain cells with different information in them in the end. And this process is known as independent assortment. So independent assortment, that's just when the chromosomes kind of align by themselves, not favoring a particular pattern, not favoring like, oh, mom in this cell and dad in the other cell. No, it could favor any number of patterns. That's independent assortment, right? So yeah, for the sake of simplicity then, let's just consider one cell to having mom's chromosomes and one cell to having dad's, right? That just makes it easier on us, right? And then we kind of proceed as normal, right? We have anaphase 1, where we're separating these chromosomes. We're separating these chromosomes. And we go into telophase 1, where we create the cleavage furrow, and we do the whole, this is like the only very normal part, where we decondense the chromosomes, and we cytokinese them. So cytokinesis, that one doesn't change. It's not cytokinesis 1, it's cytokinesis 2. We go through cytokinesis, and now we have 2 cells. But I ask the question, are these 2 cells identical? Are they? Well, no, right? We have one cell with mom. We have one cell with mom's genes and one cell with dad's genes, right? And also look, right? Once we split off, right? Once we split off, how many chromosomes do we have? Do we have four? No, no, because we separated we separated the homologous chromosomes. So now these cells are actually considered to be n equals 2. So remember, this X is only one chromosome, right? Chromatids, the chromatids have not yet been separated, right? So the X is considered to be one chromosome. So when we split them off, when we split off these cells, we are only going to have half the amount of information that we started with. So this would now be, like I said, n equals 2. So 2n versus n, 2n always is going to have the homologous pairs. N is not going to have, or 1n, however you want to say it. So, cool. Let's move on. So let's move on with this cell. So just imagine all of this is happening with both cells, because at the end of meiosis we're going to create 4 cells, right? Let's just envision this process with just one for simplicity's sake. So let's take our resulting cell, right? Let's go ahead and erase everything else, right? So now it's the same kind of song and dance that we've done before. It's basically just mitosis again. We're going to now condense the chromosomes again. We're going to deform the nuclear envelope. This is prophase 2. So after prophase 2, we're going to metaphase 2. So now during metaphase 2, we're going to align one by one. And then we're going to split sister chromatids. So we're not splitting homologous chromosomes. We're splitting sister chromatids this time, because remember, we don't have any homologous chromosomes, right? So now kind of looks like that, right? So that's anaphase 2. And then we go into normal. You go into telophase 2. We reform our nuclear membrane. We decondense our DNA. And finally, as a result of this, we go through cytokinesis, and we have 2 cells that are not identical to each other. And if you take this process and you do it again with the other set of cells, we actually have 4 cells that are a result of this process, all of which are hopefully different. Right? So during fertilization, fertilization is when we take 2 of these cells and we combine them, and now we create a zygote, which will then become a fully fledged organism. Now that organism is going to have the proper number of chromosomes. So remember, all of these chromosomes are now considered n equals 2 instead of 2n equals 4. So n equals 2 has half the amount of information that it originally did, right? Which is good for later when we want to do sexual reproduction, right? So that was the whole process of meiosis, right? So for example, we're making haploid cells. Just as a reminder, mitosis we're making diploid cells, and meiosis we're making haploid cells. This is going to, like, an example of this would be a sperm cell and also an egg cell, right? Um, One thing that you might have noticed is that we did not go through DNA replication twice. We only went through it once, and that was the original one before G2 phase. So we only go through replication once, not twice. At the end of meiosis 1, we had daughter cells with duplicated chromosomes, but we're still haploid, right? Also very important to know. And chromosomes in meiosis 1 underwent crossing over. However, it did not happen in meiosis 2. And for example, if we have an organism with 10 chromosomes normally as a diploid cell, there would be 10 chromosomes at the end of mitosis and 5 chromosomes in the end of meiosis, right? Which makes sense, right? Hopefully. So this is all for the production of gametes, and gametes are for sexual reproduction. So I would ask any questions, but I don't think there are very many to be had because I can't really hear you. So yeah, cool. Let's to finish off, we're about an hour in here. To finish off, let's start a little conversation about energy, right? So what is energy, right? Energy is the capacity to do work or make a change, right? And within energy, we have a couple of different laws, right? So we have the laws of thermodynamics. The laws of thermodynamics. Sounds really scary, I know, and they kind of are. What the hell even is a thermodynamic? So we have law number 1, which states that energy cannot be created or destroyed. It can only be transferred. So if you think of it, right, that makes sense. Have you tried creating energy? Yeah, you really can't, right? I mean, even now, just breathing in, I'm facilitating some sort of change, some sort of energy transfer, right? It cannot be created or cannot be destroyed, only transferred. And number 2 states that entropy is always increasing. So, a couple things to be said here. For one, what is energy? Well, like I said, energy is the capacity to do work or make a change, right? What is entropy? That's a little more complicated. Entropy is a measure of disorder. Right? Entropy is a measure of disorder. And what is disorder? Well, just look at your life, right? Look at how hard it is for things to kind of be kept in order, right? Think about people. How hard is it to keep people in order? Or a government? Or a society? Or beyond that, a planet, right? Entropy is always increasing. The measure of disorder is always increasing. And trying to decrease it, I mean, sure, we could decrease it for a short amount of time, but in the end, entropy is going to win, right? Everything is going to want to strive towards disorder, right? Think about a pot, right? Think about a nice little vase or a pot. Think about how much effort it took to make a vase or a pot. You had to gather the materials. That takes energy. You had to put it on the little spinny thing. That takes energy in a couple of different forms. And now, then you have to glaze it or whatever. However you make a pot, right? Think about all of that process that went into making a pot. Now you have a cat who sees that pot and thinks it'll be fun to knock it down and see it shatter. How much energy did that take? Almost none, right? Entropy is always increasing. Breaking that vase and taking the broken shards of that vase and making it into a new vase is going to be a lot harder than the actual action of breaking it, right? So entropy is always increasing. So with that in mind, how does this apply to the human body or to biological systems in general? We have metabolism, right? And before we talk about metabolism, we have to discuss a quick little equation. And I know this equation is going to be a little scary. I know it's going to be a little scary, so please bear with me, right? Um, just give me one moment. We have delta G is equal to heat, the change in heat, minus the temperature times entropy. I think that's correct. Yeah. So delta G is equal to delta H minus T times delta S. So that's a lot of terms, right? I know, it's horrifying, it's scary, I know. But please bear with me. Delta G describes the amount of free energy within a system. Now, what is free energy? Free energy is just the capacity the capacity to do work or to change something that's just kind of floating around, right? Delta H is the no, is the heat. Is the enthalpy. Enthalpy is one of my least favorite words ever. Enthalpy is something that caused me great sorrow while I was in high school AP chemistry. Enthalpy basically is just heat. Just think of it like heat, right? Makes more sense that way. Temperature temperature is of course the measure of how hot or how cold is something, and this is measured in Kelvin, right? Basically, temperature is just a measure of the average kinetic movement of a bunch of particles, right? And delta S is going to be the change in entropy, right? So this equation details. This is called the free energy equation. It details delta G, which gives free energy, right? So again, please bear with me. There's only a certain amount of information you have to know from this sort of spiel I'm giving you currently. Basically, delta G can either be positive or it could be negative, right? And what that means is when it's positive, that means a chemical reaction or a process is going to be non-spontaneous. And when it's negative, that means it's going to be spontaneous. And when something's non-spontaneous, that means it is going to be endergonic. And when it's spontaneous, it means it's going to be exergonic. So what the hell do all of these words mean? Right, so if you think of spontaneous, you think of someone who's spontaneous, right? What does that mean? It means they go out, they do stuff on the whim, right? In the same way, a chemical reaction can just be done on the whim of something, right? Spontaneous, it just kind of happens randomly. Non-spontaneous, that's when you need to put a little more energy in, right? So if you think about it like this way, right? If you think about a system, right? Think of a system like a closed, like a box, right? Now, consider this: if a reaction is going to be exergonic, if it's going to be spontaneous, if the delta G is going to be negative, right? That means the environment outside of the box is going to be the energy is going to be released into that environment. And if you think about the other way around, if it's positive, then the energy is going to be absorbed into the environment. Right? It's going to be absorbed into the box, specifically. So non-spontaneous, endergonic, you're going to absorb free energy into the system. And if it's exergonic, it's going to be spontaneous, it's going to release energy from the system. And the system in quotes, right? The system. What does that even mean? It's a thermodynamics term. It's a physics term. However, when you say it, it's a little. It changes, right? If you think about like a chemical reaction in a vacuum, that's the system, right? And outside of the system, if energy is going to be released, then we can start coupling reactions and whatnot. And if you think about it, like if you think about it within the system, then we have to take energy from outside. If it's energonic, we have to put it positive, whatever, right? It's all way too complicated, right? It's all it's all kind of its own thing, right? So yeah, like I said, if it's going to be negative delta G, it's going to be spontaneous, it's going to be exergonic, right? So, what does that mean for us? Right? What does that mean for us? Within metabolism, we have two types of metabolism. We have anabolism and we have catabolism. So anabolism is when we are building something. This is the vase. We're building the vase, right? Catabolism is when we're breaking the vase down, breaking something down. Building and breaking. Anabolism, catabolism. Put them together, metabolism, right? So something that's catabolic is going to be exergonic. And something that's anabolic is going to be endergonic. Meaning that we have to put energy into a reaction that's anabolic, and we get energy out of a reaction that is energonic. Right? I hope that makes sense. Right? So what does that mean? Right? Something like photosynthesis. Photosynthesis is the process of a plant making sugars to grow. Right? So that process would be taking CO2 and water from the air, which is which are both simple molecules, and turning that into a sugar, which is a complex molecule, right? So anabolism would be building something from nothing, basically, and catabolism would be taking something and turning that into energy, turning that into nothing and energy, right? So catabolism, we're breaking something down, and anabolism, we're building something up. Specifically, with catabolism, an example of that would be cellular respiration. So you think of cellular respiration, you take sugar, you take oxygen, you do a whole bunch of chemical reactions, and eventually you end up with CO2, you end up with water, and you end up with energy, right? So anabolism, catabolism, one is energonic, one is exergonic, one takes in energy, one releases energy, one requires more energy to be put in, one releases energy to be used. I hope that makes sense. I hope that was not just a mindless ramble about thermodynamic concepts. In any case, yeah. Yeah, so reactions, so with metabolism in mind, right, I'm going to show you a little ladder, right? So I have a perfect image here. I just wish it would be a little higher quality. So right here, right? Look at this. Look at this image. The oxidation of sugar in a cell versus the burning of a sugar. Right? Look at all these little steps. Right? Look at all these little steps versus just burning sugar by itself. Here we're kind of breaking everything down into little tiny steps. We're putting in just a little bit of energy up here at the top, but from there it's just a downhill little thing, right? Versus here, the burning of sugar, we have a big activation energy. We require a lot of flame, a lot of fire, a lot of heat, a lot of energy, and it just makes this product. And it's all this energy is released in a way that's not really used. We can't just burn sugar. We had to oxidize sugar in a way that's useful to the body, right? This would be an example of something that's exergonic, right? Specifically, or not specifically. Furthermore, right? These this would be an example of coupling reactions. So we're taking the energy from this reaction and we're using it in this reaction to proceed, and we're using that energy in this reaction, and so on and so forth, to facilitate many chemical reactions that can turn sugar into something that's actually helpful to the body. And this is called coupling a reaction, right? Coupling with something. So when you couple something, you're adding something to something else, right? That's basically it, yeah. So with that in mind, right, we could therefore couple ATP, which is energy currency, right? We could couple ATP, and we could use that as energy, right? We could break down ATP, and using the energy from that, we could put that into a process which can then proceed down the ladder. It could make more chemical reactions happen, and again and again and again. That's an example of coupling reaction. So a good molecule for that would of course be ATP, not glucose, not DNA, ATP. So that's your exam. I'm going to bed now, so have a good exam tomorrow. I'm sorry this is out so late. I had a very busy weekend. In any case, I hope you all I bid you all farewell, and I'll see you all when I see you. So, bye.